For billions of years, biology had one strict, unshakable rule. Plants capture the sun and animals don't. It was an absolute, permanent boundary of evolution. But in May 2026, scientists at the National University of Singapore completely shattered that rule. They successfully transplanted the power of plant photosynthesis inside a living animal eye. And it actually worked. This isn't just about slightly uncomfortable, scratchy eyes. Dry eye disease tortures over 1.5 billion people worldwide. It causes chronic pain, corneal scarring, and blurred vision. Inside an inflamed eye, a microscopic war is raging. The tissue gets overwhelmed by destructive particles called reactive oxygen species. Think of them as tiny molecular bullets destroying the cell's natural defenses. Current multi-billion-dollar pharmaceutical drugs only target the inflammation. They can't fix the broken cellular engine. To solve it, scientists stopped looking at medicine bottles and walked into the grocery store vegetable aisle. They took inspiration from the Sacoglossan sea slug, the only animal that steals solar power factories from the algae it eats. The slug uses those stolen chloroplasts to live off sunlight. The team at NUS asked a wild question. What if we could give humans that same ability? They took ordinary spinach leaves and extracted their microscopic power plants. They stripped away everything that consumes energy, keeping only the part that creates it. They called it leaf. At just 400 nanometers wide, these spinach-derived particles are small enough to slip directly inside a damaged eye cell. The delivery method is a simple eye drop powered entirely by the ambient light already surrounding you. When they put these spinach droplets to the test, the results shocked the medical community. Within just 30 minutes of light exposure, cellular oxidative damage tanked by 95%. In live animal trials, the leaf drops completely reversed corneal damage to near-healthy levels in just 5 days. It didn't just work. It completely outperformed Restasis, the world's leading dry eye drug. 2 months of rigorous safety testing showed zero irritation and zero toxicity. The animals moved around perfectly normally, with no idea their eyes were now operating in part like a plant. Human clinical trials are the next step, but this discovery is much bigger than dry eyes. This same cellular damage is the root cause of retinal blindness, arthritis, and muscle decay. The NUS team is already developing ways to deliver these photosynthetic molecules deep inside the human body. We've spent centuries asking what synthetic chemical we can invent. This paper asks what nature has already perfected over 3 billion years, and how do we borrow it? The next time you see spinach, remember it might just hold the future of human medicine. If you are curious about the research shaping our future, this is your space. Subscribe to ScienOMed and stay tuned.